Phospholipids. Phospholipids are compound lipids which contain phosphoric acid. That is, in addition to the alcohol, the it is connected to fatty acids, but thirdly it is connected to a phosphate group, then a nitrogen base. These are called phospholipids. There are two classes of phospholipids: glycerophospholipid and sphingophospholipid. Glycerophospholipid contains glycerol as the alcohol, while sphingospholipids contain sphingosine as the alcohol. Glycerophospholipids are the major lipids. They contain glycerol 3-phosphate, and the C1 or C2 are derived with fatty acids. So usually C1 contains saturated, while C2 contains unsaturated fatty acids. For example, we have phosphatidic acid. The phosphatidic acid here, the C1 is esterified with fatty acid 1, C2 is esterified with fatty acid 2, while C3 is esterified with phosphate group. This is the simplest phospholipid. It does not occur in good concentration in tissues, but it acts as the intermediate in synthesis of other triacyl glycerols and phospholipids. The other glycerophospholides containing different nitrogen bases or other groups may be regarded as derivatives of the phosphatidic acid. Next is lecithin or phosphatidylcholine. This is the most abundant phospholipid in the cell membrane, and it is the phosphatidic acid with choline as the base. So the lecithin has, after the phosphate group, we have the choline group attached to it, which is CH2 CH2 N CH3 CH3 CH3. There are two parts in this. The dipalmityl lecithin, it is an important phosphatidylcholine found in lungs. It is the surface active agent and prevents adherence of inner surface of lungs due to surface tension. Respiratory distress syndrome in infants is a disorder characterized by absence of dipalmityl lecithin. We don't have dipalmityl lecithin. Then second is lysolecithin, which is formed by removal of fatty acid either at C1 or C2 of lecithin. Third is cephalin or phosphatidyl ethanolamine. Ethanolamine is a nitrogen base present in cephalins. Thus lecithin and cephalin differ with regard to base. This is the cephalin. Here the nitrogenous base was choline, while here it is ethanolamine. Fourth is phosphatidyl inositol. The stereoisomer myoinositol is attached to phosphatidic acid to give phosphatidyl inositol. This is an important component of cell membrane. The action of certain hormones, that is oxytocin, vasopressin, is mediated through PI. Phosphatidyl inositol has myoinositol stereoisomer attached to the phosphate, and it acts as the it is useful in the action of oxytocin and vasopressin. That is both of these are hormones of pituitary, especially the posterior pituitary. Next we have phosphatidyl serine. The amino acid serine is present in this group of glycerophospholipids. Phosphatidyl threonine is also found in certain tissues. Plasmogens. When a fatty acid is attached by ether linkage at C1 of glycerol in glycerophospholipids, the resultant compound is plasmalogen. Here we see it is a plasmalogen. Here in the C1 we do not have an ester linkage but rather an ether linkage. Phosphatidyl ethanolamine is most important, which is similar in structure to phosphatidyl ethanolamine but for ether linkage. An unsaturated fatty acid occurs at C1, choline and inositol and serine may substitute ethanolamine to give other plasmogens. Then we have cardiolipin. It is so named as it was first isolated from heart muscle. Structurally, a cardiolipin consists of two molecules of phosphatidic acid held by additional glycerol through phosphate groups. It is an important component of inner mitochondrial membrane. Two molecules of phosphatidic acid held by an additional glycerol through the phosphate group. The cardiolipin, this is attached to C1, this is attached to C2, while in C3 we have two molecules of phosphatidic acid. Two molecules of phosphatidic acid attached through, held by an additional glycerol through the phosphate groups. This is a glycerol. So this is the primary glycerol, this is additional glycerol. This additional glycerol has phosphate attached on its C1 and C3. Cardiolipin consists of two molecules of phosphatidic acid held by additional glycerol through phosphate groups. It is an important component of inner mitochondrial membrane and essential for mitochondrial function. Decreased cardiolipin levels may result in mitochondrial dysfunction, aging, hypothyroidism, cardioskeletal myopathy, and cardiolipin is the only phosphoglyceride that possesses antigenic properties. Talking about sphingomyelins or the sphingospholipids. Sphingomyelin, the sphingosine is an amino alcohol present in sphingomyelins. They do not contain glycerol at all. Sphingosine is attached by an amide linkage to a fatty acid to produce ceramide. This is our sphingosine. It is attached to fatty acid to produce ceramide. Here, this is the attached fatty acid, and this whole group is then called ceramide. The alcohol group of sphingosine is bound to phosphorylcholine in sphingomyelin structure. This is the phosphorylcholine bound to the alcohol group of sphingosine. Sphingomyelins are important constituent of myelin and are found in good quantity in brain and nervous tissues. Ceramide was this sphingosine plus fatty acid. It acts as a secondary messenger by regulating programmed cell death or apoptosis, cell cycle and cell differentiation. A ceramide containing 30 carbon fatty acid is a major component of skin, and it regulates skin's water permeability. Talking about the functions of phospholipids, phospholipids constitute an important group of compound lipids that perform a wide variety of functions. First, they form the structural component of membranes, as phospholipids are the primary component of the membranes, and they also regulate thus the membrane permeability. Phospholipids, lecithin, cephalin, cardiolipin in the mitochondria maintain conformation of electron transport chain and thus the cellular respiration. Phospholipids participate in absorption of fats from intestine. Phospholipids are essential for synthesis of different lipoproteins and thus participate in transport of lipids. Accumulation of fat in liver can be prevented by phospholipids, and hence they are regarded as lipotropic factors. Arachidonic acid, an unsaturated fatty acid liberated from phospholipids, serves as a precursor for synthesis of eicosanoids, and phospholipids participate in the reverse cholesterol transport and thus help in removal of cholesterol from the body. Phospholipids act as surfactants, that is, agents lowering the surface tension. For example, diapalmitol phosphatidylcholine is an important lung surfactant. Respiratory distress syndromes in infants associated with insufficient production of this surfactant. Then we have cephalins, an important group of phospholipids participate in blood clotting, and phosphatidyl inositol is the source of second messenger inositol triphosphate, which are involved in action of the oxytocin and vasopressin.